Hello students, welcome to this session. In this session, we are going to be solving the questions from AIET 6 examination, and let's get started. So the first question we have is question number 108, pyramids of number is. So our options are always upright, always inverted, and then either upright or inverted, and then neither upright nor inverted. So in this question, when we talk about the pyramid of number, so we understood that the flow of energy happens based on a particular ecosystem. So let us take a particular ecosystem such as a forest. Okay, so we have forest as one of the example, and then let us take a tree as another example. So when we talk about the number means, that, for example, if I take in a forest, if you just generally analyze, we see that the producers, I'll write it as P. So they're very high in number, correct, right? So you can see that all the trees and all the plants and shrubs, everything that are photosynthetic, they're very high in number in a forest ecosystem. And then follows the lesser number, that is herbivorous comes next, correct? And then carnivorous, and then we have omnivore or something like that. So now the pyramid is represented in such a way that you can see it's a pictorial representation where the highest number of individuals in an ecosystem, such as grassland or a forest ecosystem, the highest would be the producers. And the second would be the consumers, where the herbivores come. And next would be the carnivore, where they occupy a lower position. So it's based on the numbers, correct, right? So here the pyramid is upright. We call this pyramid as upright. Okay. Whereas if I take tree as an example, the herbivore, the producer, is only one tree, whereas the rest of the colony that depends on this tree. For example, there might be insects such as cedars, which you can hear the sounds, chirping sounds. Okay, the birds who build their nests, the small birds who feed on these insects, or the large birds that feed on these birds. So here, what happens is the number of consumers, they occupy the highest position. Okay, such as the birds or insects, let's say, and then comes the birds, the large birds, and then comes the small birds, and then comes the large birds, and finally it is one tree. So what can you see here? The pyramid is inverted when it comes to such a situation. So according to our question, the pyramid of number is, it can either be upright or it can be inverted. So this is called as an upright pyramid, and this is called as an inverted pyramid. So if you take two situations as an example, so our correct option would be option C, that is either upright or inverted. Okay, so let's go for the next question. And the next question is 109, and the statement 1 says, nuclear envelope remain intact throughout the prophase. And the second statement is, at the end of prophase, when viewed under the microscope, a cell shows distinct Golgi, endoplasmic reticulum, and nucleolus. So let's see what are our options. Statement 1 is correct, but the statement 2 is incorrect. Statement 1 is incorrect, but the statement 2 is correct. Statement 1 and the statement 2 are correct. And then we have both statement 1 and statement 2 are incorrect. So we know that during the cell division, it has different phases. So we divide these phases as prophase, metaphase, anaphase, and telophase. So the question is based on the prophase. So here it is clearly mentioned. What happens in prophase? So when we look at a cell which is entering the prophase, which is the first phase of the cell division, so here in the prophase, the first thing that happens is all the chromatin fibers, okay, they get completely loosened up. So the nucleus becomes, quite condensed. We can call it as condensed. So as it condenses, okay, first the nuclear material gets condensed. Second one is the nuclear membrane slowly starts disappearing. Okay, so by the end of the prophase, you can't even see the nuclear membrane. So nuclear membrane, I'll write it here. Okay. nuclear membrane, they disappear. Next one is the important organs like Golgi, cell organelles, Golgi, endoplasmic reticulum. Okay, so all this will start disappearing by the end of your prophase. So what remains in the end of the prophase is you can see that the nuclear material has gone into high levels of condensation. Okay, and disappearance of nuclear membrane will take place. Disappearance of cell organelles like Golgi apparatus and endoplasmic reticulum, as well as the nucleolus, happens during the prophase. So the statement one says nuclear envelope remains intact throughout the prophase. That is actually wrong. The first thing that happens is disintegration of the nuclear membrane, and the second one is at the end of prophase, when viewed under the microscope, a cell shows distinct Golgi, endoplasmic reticulum, and the nucleolus. So as we discussed just now, both of the statements are wrong. So our option is both statement one and the statement two is incorrect. Okay. So let's go for the next question. And the next question is we have to match list one with the list two. In the column one, we have dodo, and then we have quagga, we have thylacine, and then we have a stalaco. So these are, and then we have the column two, such as Africa, Russia, Mauritius, and Australia as the list two. So we have to match which of these endemic species belong to which region. Okay. So let's look at this. When we talk about dodo, which is a bird, which is it appears in Mauritius. So I will write it as C. Okay. And very easy to remember. Stelaco goes for Russia. So I'll write it as 4. Okay. I'll write it as 3. Okay. It goes for Russia. Now, these two are done. And then when we look at this quagga, and then we take a look at thylacine. So when you see quagga, when you see quagga, quagga actually belongs to Africa. Okay. So I'll write it as here for quagga, Africa. And then thylacine belongs to Australia. So I'll write it as C. So it's, thylacine belongs to Australia. So this goes C. Okay. So dodo is Mauritius. I'll write it here A. Okay. And then this is D. And this is B. And okay. So where is our option? Our option appears to be option C. So again, let's see. So these are the native birds to a specific region. So dodo belongs to Mauritius. Quagga belongs to African continent. Thylacine belongs to Australia. Stella cow belongs to Russia. Okay. So let's go for the next question. So that was option C. So 111th question. Connecting link between the glycolysis and the Krebs cycle is. So our options are oxaloacetate, then we have phosphoenol pyruvate, and then pyruvate, and then we have acetyl CoA. So connecting link between glycolysis and Krebs cycle. So what happens in glycolysis, students? So we know that it is an anaerobic process. C6H12O6, which is the glucose, is broken down into two molecules of pyruvate. Okay. So this pyruvate, okay, actually before going into the Krebs cycle, it is converted into acetyl CoA. As it is converted into acetyl CoA, it enters the mitochondria, and then in mitochondria, the Krebs cycle will take place. So this pyruvate is converted into acetyl, okay, acetyl CoA, and this acetyl CoA enters the mitochondria, okay. Okay. This enters into mitochondria, and mitochondria, what happens? The Krebs cycle will take place. Okay. So this conversion of acetyl CoA will happen in the cytoplasm. So this acts as a connecting link between the glycolysis as well as your Krebs cycle. So our option is acetyl CoA, which is option D. So let's go for the next question. In the next question, 112th question, differentiation of shoot is controlled by. So the first option is higher gibberellin to cytokinin concentration. Next, high auxin to cytokinin concentration. Next one is high cytokinin to auxin ratio. Okay. Next one is high gibberellin to auxin ratio. So when we talk about differentiation of shoot, usually the shoot differentiation happens with less concentration of auxin. Okay, and then usually this is the natural way that happens. Usually the auxin concentration has to be less, and the shoot differentiation is mostly influenced by the concentration of cytokinin. So we need more concentration of cytokinin and the less concentration of auxin for the promotion of the shoot differentiation. Now, what happens is during the tissue culture process, when we are doing in an artificial medium, so basically we balance the ratios between the cytokinin as well as the auxin concentration, where the cytokinin concentration will influence the plant in increasing its shoot growth, whereas the auxin concentration will influence the root growth. So shoot and root, you can understand, right? So that is why when we talk about differentiation of shoot, the shoot is controlled by high amount of cytokine should be there and low amount of auxin should be there. So our option is C. Okay, next one. So 115th question. Cross between AABB and AABB will form. So we have these are the alleles which are given to us. So let's see what happens if we conduct a cross between them, and here are the options. So if I take one parent as AA as given in the question, AA, and then BB is crossed with small a, capital B, correct, right? So now what will be the gametes? So these are the parents. Okay, so let us see what type of gametes will be formed. So here the two types of gametes will be formed. One is capital A, capital B, which is one type of gamete, and the other one is small a and capital B. This is another type of gamete, correct? And here, what would be the possible gametes? We have same alleles. Okay, so we have only one type, that is capital A and capital B as the type of gametes that would be produced in the other parent. So let's see what happens here. So if these go, I mean, like the possibility of getting these gametes would be, so let's say that this gamete goes with this. So let's see, A, B, B, correct? And if this gamete fuses with this gamete, what would we get? We get small a, capital B, capital B. What did we get, students? We have capital A, small a, capital B, small a, small b, small a, capital B, capital B. So do we have an option? Yes. See, we have one. Let us say this is the F1 generation. In the F1 generation, what do we get? We get capital A, capital B as one. And the other individual would be small a, capital B, capital B. Got it? So this question can be answered by just observing what are the parental alleles or the genotype of a parent. So if you look at the genotype of the parent and properly gather what type of gametes would be produced, the probability of you getting the answer right would be very, very high. Okay, let's go for the next question. Our next question is 117th question. Parthenocarpic tomato fruits can be produced by. So our option is treating the plants with low concentration of gibberellinic acid and auxins, and then raising the plants from vernalized seeds. Next one is treating the plants with phenyl mercuric acetate. And the final option is removing androecium of flowers before pollen grains are released. Very interesting question. So first of all, what is parthenocarpy? We know that the parthenocarpy is we can just translate it to parthenogenesis. So whenever the fruit formation takes place without fertilization, okay? So without fertilization, that is called as parthenogenesis, correct? So whenever the parthenocarpic fruit is born, it is a seedless fruit. For example, you might have heard about grapes, which are the seedless grapes, correct? We use it maximum. So how do we induce parthenocarpic? Usually the parthenogenesis can be induced artificially as well as it can be induced naturally. So now what happens during parthenogenesis? How does the parthenogenesis occur? The most common factors that affect the parthenogenesis is the concentration of gibberellinic acid and the auxin. Whenever we present the plant with low concentration of gibberellinic acid and auxins, so that will induce parthenogenesis. Now, in what type of plants it can be induced? See, this should be done in an unfertilized flower. That means when the flower is there, and let's say we have androecium and we have gynoecium, correct, right? So androecium represents the male parts and gynoecium represents the female parts. Now, if we have flower in a garden, what we have to do is we have to emasculate. Emasculation means remove all the anthers before maturity so that we are left with only gynoecium. In such plants, when we induce low concentration of gibberellinic acid and auxins, so that will result in parthenogenesis. And why did we emasculate? In case if it just fertilizes before this is done, then the parthenogenesis will fail. Understood? So that is why this option is absolutely right. Coming to the other options, other options might confuse you, because of the words or because of the confusion in the concept. So let's look at them. Raising the plants from vernalized seeds. What is vernalization? Vernalization is a process of breaking seed dormancy. So what do? We are breaking seed dormancy. How do we do? We break the seed dormancy by chilling the seeds. Okay. When we take the seeds and we chilled seeds are taken out, and when they're put in normal conditions, then the seed breaks its dormancy. That is called as vernalized seeds. Okay. Next one, treating the plants with phenyl mercuric acetate. So phenyl mercuric acetate is actually an anti-transparent. Okay. Have you heard about transparent, anti-transparent? What is transparent? Transpiration is a process where the water is lost through the stomata during the daytime, correct, right? So anti-transparent are sprayed on the plants so that we close the stomata, or we induce a closing of the stomata such that the loss of water is prevented. Okay, so that is called as anti-transparent. An example for an anti-transparent is phenyl mercuric acetate. Coming to the final option, removing androecium of the flower before the pollen grains are released. This is called as emasculation process. Okay, so these are the options, and our right option for this question is option A, as we have seen. So let's go for the next question. And our next question is, what would happen if in a gene encoding a polypeptide of 50 amino acid, okay, 25th codon is UAU is mutated to UAA. Okay, very interesting question. And our options are a polypeptide of amino acids will be formed. Next one is two polypeptides of 24 and 25 amino acids will be formed. Next one is a polypeptide of 49 amino acids will be formed. And then finally, we have a polypeptide of 25 amino acids will be formed. This is a very close question because most of you can get it wrong. Just we have to stick to the basics. So what I have the given, let us say that we have a polypeptide chain. And in this polypeptide chain, it starts from 1 and it ends at 50. Correct, right? So these are the number of amino acids that could be produced in a gene. So let's say it's an mRNA. But on the 24th, let's say that in the 25th amino acid, what do we have? We have UAA. Correct, right? We have UAA. Now, what happens is, in the first amino acid, we know that we have to have a start codon, which is, AUG. Now, from here to here, it goes smoothly, and the 25th codon is UAA. What is UAA? UAA is a nonsense code. Okay? Nonsense code. So when we have nonsense code, what does it lead to? Nonsense code will lead to the termination of our entire translation. So here we have the translation bubble. It keeps on synthesizing the polypeptide chain 1, 2, 3, 4, 5, 6, and all the way it goes till the 24th amino acid. Okay, it goes all the way till the 24th amino acid. But when it comes to the 25th amino acid, what happened? It leads to termination because it encounters the stop codon or the nonsense codon. So what do we have here? From 1 to all the way till 24 amino acid, it was uninterrupted. But at the 25th, it was stop codon, so the translation will stop. What happens to the rest of the codes? The rest of the codes cannot be translated. Why? Because once a termination is completed Again, to begin the translation, again, you need a start codon, and we don't know what codon we have on the 26th amino acid, correct, right? So that is why the rest of the gene will get degenerated, and we do get a polypeptide chain of 24 amino acid sequence, okay? We get 24 amino acid sequence of polypeptide chain. So look at these options. A polypeptide of amino acids will be formed. Yes, correct. Two polypeptide, 24 and 25 amino acids will be formed. As I told you, 24 is possible and 25 is not possible. So this is wrong. Next one, because they have given two polypeptide chain, so the option is wrong, okay? Next one, a polypeptide of 49 amino acids will be formed. This is also wrong. Why? Because it is terminating in the middle. A polypeptide of 25 amino acids will be formed. This is also wrong because the last codon, especially the UAE codon, do not code for any amino acids. So when it does not code for any amino acid, 25 amino acids is impossible. So we can get only 24 amino acids. Okay, so let's go for the next question. Pteridophytes differ from mosses or bryophytes in possessing. So independent gametophyte, then we have well-developed vascular system, then archegonia structure, then we have flagellate, spermatozoids. So pteridophytes differ from bryophytes. So when we look at a pteridophytes, one of the major difference that we can see is a pteridophyte body, plant body, is a sporophyte. Okay, plant is sporophyte. That means it can be divided into leaves, shoot, and root. Correct. So that is one thing. Second thing that we can see in the pteridophytes is they have well-defined vascular system. So that we cannot see in the bryophytes. They are gametophytic and the vascular system is very, very poorly developed or it is absent. So that is what makes a very big difference. So pteridophytes differ from bryophytes in having well-defined vascular system. Understood? Yeah. Let's go for the next question. So which of the following shows the concept of species area relationship? So 130th question. So let's see the options here. The number of species in an area increases with the increase in the size. Okay. Larger species require larger habitat areas than the smaller species. More species within any given area are endemic. And the larger the area, the greater the extinction rate. So this is the principle of Hardy-Weinberg principle. So he talks about species area relationship. So why did we get here? So it says that as we keep on exploring the larger areas, we encounter new species. The possibility of encountering new species is very prominent, correct, right? For example, if you take Amazon, very little bit part of Amazon is explored. Now, if we keep on expanding our research and keep on searching for newer habitats, what do we encounter? We have the high possibility of encountering new species, new organisms, correct, right? And is it forever going on? Definitely not. So now our search, when we start, it starts from a little bit of species, and as we increase our search, the number of species keep on increasing as we encounter new organisms. But this is not like it can happen forever. At one point, this will stop. Okay, so at one point, it has to stop. So he says that the number of species in an area increase with the size of an area up to a certain time, up to a certain point. After that, it will stop. So our correct option for this is species area relationship is option A. So let's go for the next question. Question 133. Statement 1. Increase in the mass and area in number of individuals are twin characteristics of growth. Metabolic reactions can be demonstrated outside the body in an isolated cell-free systems. In the light of the above statements, choose the correct answer for the given options. Statement 1 is correct, but the statement 2 is incorrect. Statement 1 is incorrect, but the statement 2 is correct. Both statement 1 and statement 2 are correct. This should be 2. Okay. Next one. Both statement 1 and statement 2 are incorrect. So let's take a look at the first question, first statement. Statement 1 says increase in mass, correct? and increase in number are twin characteristics of growth? Definitely yes. If we look at larger organisms like multicellular organisms, we look at the mass. So you can see that in your younger age, they would be measuring your height or they would be measuring your weight. And they would tell, okay, you have put on weight or you have lost weight or you have grown tall. So these are the characteristics of growth. Whereas when you look at the number can be very clearly understood by applying it to the microorganisms. So if I take, let's say, 100 microorganisms and then grow it in a cultural medium, so after a few days, I can see that they have divided and they have increased their colony numbers. So we can take number as a property of growth or as a criteria of growth when it comes to such unicellular organisms. So they become a twin character for growth. Now, statement 2. Metabolic reactions, yes, can be conducted in a cell-free medium. Okay, that is outside the cells in a laboratory, we can do it. Okay, so both the statements appear correct. So we go for statement 1 and statement 2 are correct. So our correct option is option C. So our next question is, which of the following is a characteristic feature of Fabaceae? So which of the following is not the characteristic feature of Fabaceae? Let's take a look at the option. The first option is the root system, the compound leaves and racemes, taproot system, compound leaves and racemose, raceme inflorescence. And then we have flowers are actinomorphic, twisted estivation. And then we have gamopetalous. Then we have stamens are 10, interrose, basifixed, and dithecous. And finally, we have monocarpellary ovary superior and then bent stigma. A lot of characteristics are very relevant to Fabaceae or their Fabaceae characteristics. For example, if you look at Fabaceae, yes, they exhibit taproot system. You can just remember so many characteristics with this question itself. They have taproot system. They exhibit the presence of compound leaves and they have racemes inflorescence. Clear? Next one. Coming to our flowers, the flower are actinomorphic. So it is zygomorphic. Okay. They are zygomorphic. Twisted estivation. It is imbricate estivation. Okay. So it is given gamopetalous. It is actually polypetalous. So this option appears wrong or it is wrong. Okay. Whereas if you see the other options such as stamens at 10, there is interrose, they're basifixed and they're dithecous. Correct. This option is right. Next one is monocarpellary ovary, correct. Ovary superior and the bent stigma is bent. Even this option is correct. So our wrong option for 137 is B. And I've given you, it's not actinomorphic, it is zygomorphic, it is not twisted, it is imbricate, it is not gamopetalous, it is polypetalous. Clear, people? So this is for 137th question. Let's go for the next question. And our next question seems to be match the following. So our column 1 has disintegration of nuclear membrane, appearance of nucleolus, and then division of centromere, replication of DNA. And our column 2 has anaphase, prophase, telophase, and S phase. Very, very easy question, students. You need not go in an order, mix up your order, and see which is very relevant. So let's take one of the easiest thing, that is replication of DNA. When does the replication of DNA happens? We know that before the cell enters into the cell division, it has to go through this replication of DNA phase. So if I take the cell cycle, we know that we have growth 1 phase, and then we have S phase, and then we have G2 phase, and then we have M phase, and then finally we have G0 phase. Correct, no, people? So in the S phase, what happens? You have your DNA replication happening. Clear, this is the S phase. Here is where it is happening. So see here, there is a lot of time or there is a lot of gap between the S phase and the M phase. So that is why it's very easy to remember. Replication of DNA happens during the S phase. So I'll write it as D. Okay. This is very, very easy. Next one, disintegration of nuclear membrane. So this is the first thing that happens when the cell starts dividing. So the first thing that happens when I say, that means the first step of the cell division, especially that is prophase, metaphase, anaphase, and telophase are the steps of cell division or the phases of cell division, we can tell. So disintegration of nuclear membrane, when I say it's one of the first things that happens, that means it has to happen in the prophase. Okay. Next one, appearance of nucleolus. So when the appearance of nucleolus has to happen, that means, see, the nucleolus disappears, the chromatin material get condensed, and finally they get separated and they go to the daughter cells. And what happens when they go to the daughter cells? Again, decondensation happens and appearance of nuclear membrane happens. So this has to be the lastest phase of the cell division. So which is the last phase of cell division? Telophase. So we'll write appearance of nucleolus will happen at the telophase. And finally, we have division of centromere. Division of centromere happens especially in the anaphase. Okay, so I'll write it as C. So what is our option? So we have, let's say A is 2, yeah, A2, B is 3, C is 1, and D is 4. So look at here, option A seems to match everything. So correct option for this question is option A. So let's go for the next question. And our next question is statement 1. In numerical taxonomy, observable characteristics are not given equal importance. More than 20 characteristics cannot be studied at the time in numerical taxonomy. So let's choose the right option. Again, they have given 4 statements, okay? And the statements would be either 1 is correct, 2 is wrong, or 1 is incorrect, 2 is correct. Both the statements are correct, or both the statements are wrong, correct? So let's look at the first statement. In numerical taxonomy, observable characteristics are not given equal importance. No, students, what happens in numerical taxonomy is observable characteristics are given the highest importance. Okay, so when when we have observable characteristics, what we do is certain codes are given. Okay, we give certain codes and certain numbers. Okay, or identification is given to those observable characteristics. So this thing is wrong. Let's look at the second question. More than 20 characteristics cannot be studied at a time in numerical taxonomy. In numerical taxonomy, hundreds of characteristics can be studied simultaneously. Okay, so this is also wrong. So both statement 1 and statement 2 are incorrect. So option is option D. Let's go for the next question. Yeah, our next question is 141. Consider the following statements concerning food chains. Removal of 80% of the tigers from an area results in great increased growth of vegetation. Removal of most of the carnivores result in an increased population of deers. The length of food chains is generally limited to 3 to 4 trophic levels due to energy loss. The length of food chains may vary from 2 to 8 trophic levels. Which of the above statements are correct? 2 and 3. Next we have 3 and 4. Next we have is 1 and 4. And then finally we have 1 and 2. So we have to choose the correct statements. Let us look at the first statements. Removal of 80% of the tigers in an area results in great increase of vegetation. Definitely wrong. So I would like to tell one of the statements which read in the newspapers, that disappearance of the tiger in Karnataka resulted in drying of Kaveri River. I know it's a unusual statement to just make, but I read it in a paper. So why did we do that? When the tigers are removed, which are the apex predators in a tropical ecosystem, so when we remove them, now the herbivore population will increase at a very alarming rate. So when higher herbivores are increasing at an alarming rate, now what happens? Their consumption depends totally on the producers. So that which are, the producers actually they depend on them, and the producer population decreases very drastically. Okay, and it can lead to desertification if it is not checked. This has happened in Yellowstone National Park in USA, where the wolves were removed and then herbivore population increased very tremendously, and finally it led to the death of Yellowstone, almost to the death of Yellowstone National Park, and eventually they had to introduce around 18 packs of wolves into this Yellowstone. Within 5 years the park was back to life. So it's a very fantastic example to take. So removal of 80% of the tigers from the from the region will not increase the vegetation, but it will reverse it. So our option 1 is wrong. Removal of most of the carnivores resulted in increasing population of deer. So this is a possibility, extremely happening possibility, or 100% possibility that the population of deers will increase very exponentially if the tigers are removed. Okay. Next one, the length of the food chains is generally limited to 3 to 4 trophic levels due to energy loss. So this is absolutely right. Why? Because we can't have too many trophic levels because we have a 10% rule. So in 10% rule, what happens is we have producers, and producers which use the sunlight, they can only tap 10% of the sunlight energy, and they store it in the form of biomass. And then it is going for herbivores, they get 10% of what the producers have eaten. And then we have carnivores get 10%. So that's why you can see that the producers are the highest number of individuals on this planet. Then comes the herbivores, and then comes the carnivores. So that is because of the loss of energy from one trophic level to another trophic. So when we have a constant loss in the energy from one trophic level to another trophic level, what is the consequence? We can't have an ever increasing trophic level. So it has to stop at one point. So that is why we have all the the trophic levels are limited to either 3 to 4 levels, not more than that. Okay, this option is also correct. Next one. The length of the food chains may vary from 2 to 8 trophic levels. So this is wrong. Why? Because the length of the food chain is limited to trophic levels. It's a food chain goes with the producers, which produce a food through sunlight, and then goes to the herbivore. Herbivore eat the producers, and then a carnivore eat a herbivore, and then we have higher carnivores where they feed on other carnivores. So this is, and then we have a detritus chain. So the food chain is limited to 4 trophic levels or 3 trophic levels at the maximum. So it cannot go. So our option, that is the statement 2 and the statement 3 are correct. So that goes with option A. So our option A is correct for question number 141. So let's go for the next question. So here we have match the following. We have list 1 and list 2. So let's see what are those. So in the column 1, we have trichoderma, streptomyces, nitromonas, and lactobacilli. And then we have column 2, nitrification, biocontrol agent, lactic acid, source of antibiotic. So when you look at these options, we have to say which microorganism is used for what purpose. So if you see trichoderma is an excellent biocontrol agent. So let's go for A. Okay. Next one. Streptomyces is used for the production of antibiotics. So I'll write it as B here. Coming to nitromonas is a nitrified. See, the name itself says you can get two things easily, very easily in this match the following. Okay, nitrosomes, nitromonas are used as nitrification bacteria. So I'll write it as C here. And then see, lactobacilli and lactic acid is so verbal. So we can just go for this. Lactobacilli that is used in the curd is used for lactic acid production. So here it is. So what do we have? 1C. We have it. Yeah. A is 3. B is 1. Correct. Strepto minus B is 4. Yeah. B is 4. Where do we have? Yeah. A is 2. B is 4. C is 1. And then D is 3. So this is the right option. That is option B is correct for 142 question. Okay. So go for what you know first, and then the rest of the options will fall in place. Some of the times, it can help, like you can tell 80-20, it can come. So just focus on what's correct first, and then we can go for what's not. So question is 148, a somatic cell that has just completed the S phase of its cell cycle as compared to the gamete of the same species, okay, has same number of chromosomes, but twice the number of amount of DNA, whereas twice the number of chromosomes and 4 times the number of DNA. And then we have 4 times the number of chromosomes and twice the number of DNA. And then finally, we have twice the number of chromosomes and twice the amount of DNA. Guys, this question is quite interesting, even though it's quite easy. Why? Because they see a somatic cell. A somatic cell is a diploid cell. So I'll write it as 2N. And then it has finished the S phase. That means it has finished the DNA replication process. And finally, we are comparing it to a gamete. A gamete is haploid and it is N. Remember that. So that is where the question entirely changes. Okay. So if a somatic cell has entered the S phase and it has completed the S phase, that means it has finished its DNA replication. If it has finished its DNA replication, what happens is the 2C, let us say that this is the total amount of DNA that was present before the S phase. So finally, it will become 4C, that is doubled, right? DNA replication means it will duplicate. But the chromosome number, let's say that in the beginning of the S phase, we have 2n, and then finally, by the end of S phase, we have 2n only. Why? Because the chromosomes are not yet separated. Okay, it's not yet segregated. Okay, whereas we're comparing to gamete. So when we compare it to the gamete, you can just tell, yes, gamete has 2n, so we have 2n here. A gamete has 2C of DNA. Okay, see here. Oh, sorry. It has one copy of DNA, right? So you can see here twice the number of chromosomes and then 4 times the number of DNA. See, 4C and 1C and N and 2N. So this is why our option B is correct. Okay, just look at the question carefully because you know the answer and you might get it wrong because you're in a hurry. Again, we have one more match the following, very interesting one. Question number 154. We have column 1 and column 2. Column 1 says fast muscle fiber, slow muscle fiber, actin filaments, and sarcomere. And then column 2 says myoglobin, lactic acid, contractile unit, and then I-band. So if you look at this question, it's quite sensible. We just have to think a little bit. So let's see what are the first things that we can answer. Sarcomere, very easy. In a muscle fiber, one of the most important region in the muscle fiber is the sarcomere. Sarcomere, you can tell that it's a functional unit of a muscle fiber. So what is sarcomere? It represents the contractile unit of a muscle fiber. So I'll write it here. So let us look at the other options such as fast muscle fiber. So whenever you have a fast muscle fiber, what happens is they rapidly burn the oxygen. That means they require high amounts of oxygen supply. But can we do that all the time? It's not possible in a living system. So what happens if, for example, you're exercising or you're running and you're doing very high intensity workouts? So what happens? You're doing a lot of muscle contraction at a very rapid pace than a usual thing. So that results in consumption of all the oxygen in the tissues, and eventually it enters into an anaerobic respiration where lactic acid accumulation will take place in the fast muscle fibers. So that leads to the cramping. So usually it happens in the fast muscle fibers, especially the skeletal muscles. So I'll write it as A. Whereas when you look at the slow muscle fibers, such as your digestive system, okay, very beautiful example is your digestive system. You will never hear of your heart cramping. You will never hear of your digestive system cramping because they are made up of smooth muscles. And the smooth muscles are actually slow muscle fibers because they know they have a very, very, very decent pace of contraction. So that will not lead to the lactic acid formation or the cramping. So that is why we can just tell that slow muscle fibers are very important fibers with respect to not cramping. And the reason for slow muscle fibers not cramping is because they have very high amount of myoglobin, which supplies oxygen to these tissues at a very high pace. So that is also another reason. So I'll write it as B here. Okay. And finally, we have our actin filaments. So we know that we have two filaments. One is actin and myosin filaments. So actin are represented something like this, right? And then myosin is something like this. Correct? So we have a myosin head and a myosin tail, and then we have an actin. So actin is also called as isomeric band or I band. Why? Because actin forms this isomeric polymer. Okay, so that is why we call it as I band. So I'll write it as C. So we have B, A, D, C. So what is the answer? A is 2. A is 2. Okay. B is 1. C is 3. Yeah, C is 4. Yeah. A is 2, B is 1, C is 4, D is 3. So our option is option C for the question number 154. Okay. So let's go for the next question. Our next question is 158. And the statement 1 says acetylcholine is released when the neural signal reaches the motor end plate. And then our statement 2 says muscle contraction is initiated by signals sent by the CNS via sensory neuron. So obviously we have 4 statements as usual. 1 is correct, 2 is wrong, 1 is incorrect, 2 is correct. And then finally we have 1 and 2 are correct, or both 1 and 2 are wrong. So let's look at the first question. Acetyl-CoA, okay, acetylcholine. It's a very important factor. Now, what happens is, let's say that this is the synaptic end, this is the NMJ, neuromuscular junction. Usually, these neurotransmitters they are released in the form of acetyl choline. Okay, so this acetylcholine, which are released, they're released at the end plate and they go and stick on to the receptors. Usually these signals are received by the receptors, and then finally you have the acetylcholine, and then they interact with the primary and the secondary messengers that leads to the muscle contraction. So acetylcholine is released when the neural signal reaches the motor end plate. This is absolutely right. Okay, this happens. Whereas when it comes to the statement 2, muscle contraction is initiated by the signals sent by CNS via sensory neuron. The statement 2 looks almost correct. Whereas if you look at the last part, it is not sensory neuron, it is motor neuron. Okay, I'll write it here. It is motor neurons. Motor neurons are the ones that carry the signal from central nervous system to our NMJs or neuromuscular junctions. So that is why our statement 1 is correct and the statement 2 is incorrect. So let's go for option. Wait. Statement 1 is correct, but the statement 2 is incorrect. Option A. Okay, so let's go for the next question. Our next question is, which ion is essential for muscle contraction? Very easy. Na plus, K plus, Ca2 plus, and then we have chlorine. Okay. So whenever we talk about muscle contraction, when we see the structure or when we observe the characteristic feature of a muscle fiber, we come across a very, very peculiar cellular organelle called as sarcoplasmic. If you remember this, it becomes very easy, okay? Sarcoplasmic reticulum. So sarcoplasmic reticulum is a type of endoplasmic reticulum you can find in muscle fiber. Okay. What is the role of this? They act as bank for calcium ions. Okay, they act as a bank for calcium ions. Why calcium ions are required? Calcium ions are required for formation of actin and myosin complex. Okay, so to form this, to increase the affinity between the actin, you know that, right? Actin and the myosin will combine to form, they come and interact together. So this interaction happens with the help of calcium ions, which increases the affinity between actin and myosin. Without the calcium ions, you cannot expect muscle contraction to be happening. Okay, so that is why calcium ion is very, very important factor, which is essential for muscle contraction. So let's see the next question. Our next question is, fetal ejaculation reflex in human female is induced by release of oxytocin from pituitary. Next one is fully developed fetus and placenta, differentiation of mammary glands, and finally we have pressure exerted by the amniotic fluid. So fetal ejaculation reflex is occurs when a fetus is completely developed. When the fetus is completely developed, now what happens is it's a neuromuscular signal. Okay, so we call it a neuroendocrinal signal. Yeah, that's the right word. So when the neuroendocrinal signals originate from the baby and the placenta, it reaches the pituitary gland, and finally it results in the release of oxytocin. So the signals originate between the interaction that happens between the placenta and the fetus. Okay, so our option B is correct. So this should be an easy question because it's in the syllabus and in this current year, so it should not be a big problem. So let's see the next question, 174. The digestive system in platyhelminthes has only a single opening that serves as both mouth and anus. Okay. Statement 2 says organ level of organization is exhibited by the members of platyhelminthes. In the light of the above statements, choose the correct options from given below. So obviously we have 4 statements which talk about which statement is correct, which statement is wrong. So let's look at the first statement. The digestive system in platyhelminthes has only single opening. We know that it's a parasite and it has only single opening, right? And it serves as both mouth and anus. Absolutely right. Absolutely right. Okay. So let's look at the second question. Organ level of organization is exhibited by the member of platyhelminthes. True in the light of above statement. So see, when we look at the characteristics of platyhelminthes, it's so easy to remember because it's a parasite and it is also called as liver fluke. And we know that it has only one opening. We would have written the diagrams of platyhelminth as something like this. Correct, right? So we write it as something like this. And then we represent it as, okay, cloaca, mouth and anus are the same end, correct? And then usually these are organ level of organization. So both statement 1 and 2 are correct. Okay. So where is it? Yeah, it's given. Yeah, both statement 1 and 2 are correct. Okay, option C. Let's go for the next question. Myoglobin is present in all muscle fibers. Okay, white muscle fibers only, red muscle fibers only, and then we have both white and red muscle fibers. See, myoglobin is a pigment that supplies oxygen to all the muscle fibers. Okay, so when you talk about myoglobin, myoglobin is found in all muscle fibers. Okay, so that is why our option is correct. Myoglobin is something like our hemoglobin, which supplies oxygen to our tissues, especially muscle fibers, to support contraction and relaxation. The most active phagocytic white blood cells are neutrophils and monocytes, neutrophils and eosinophils, and then lymphocytes and macrophages. Okay, and finally we have eosinophils and lymphocytes. So when we talk about phagocytic activity, phagocytosis is, for example, you have an antigen. Let's say it's a bacteria. Now, what does the white blood cells do? White blood cells, what they do is they come and they engulf this parasite or any antigen and they digest within the cell and they eliminate it. So that mostly happens with neutrophils and monocytes. Neutrophils and monocytes are highly phagocytic in nature, whereas here you can see a lot of odd options such as eosinophils are non-phagocytic, lymphocytes are non-phagocytic. Again, here eosinophils and both lymphocytes are non-phagocytic. That is why our option A is correct. When it comes to phagocytic white blood cells, it is actually neutrophils and monocytes. Clear? So let's go for the next question. 186. Which of the following changes occurs in the diaphragm and intercostal muscles when the expiration of air takes place? Very beautiful question. Okay, our options are internal intercostal muscles relax and diaphragm contracts. External intercostal muscles and the diaphragm relaxes. Next one is internal intercostal muscles contract and the diaphragm relax. And then we have external intercostal muscles and the diaphragm contract. When we talk about the inspiration and the expiration process, it happens with the coordination of many tissues that are within our thoracic cavity. So just imagine our ribs, and just below our ribs is a dome-shaped structure called as a diaphragm. Now, diaphragm acts like a pump or a piston. See, this dome-shaped structure, which is just, this is the rib cage. Okay, this is the rib cage. What happens is it contracts and moves downward. When it contracts and moves downward, now it is pulled, right? And second thing, what happens? This is one rib, let's say, and this is another rib. So between these ribs, we have a muscle called as intercostal muscles. If it is towards the inside, it is called as internal intercostal muscles, that is towards your lungs. If it is towards your skin or outside to your chest, that is called as external intercostal muscles. During inspiration, what happens is the diaphragm contracts and the external intercostal muscles also contract, moving the thoracic cavity, okay, up. See, what happens, it goes downward, and see, when it is pulled out, the thoracic volume will increase. When the thoracic volume will increase, and with the external intercostal muscle support, now your ribs will move upward and forward, increasing the thoracic space. That leads to the entry of air into our lungs. Now they're talking about expiration. So now in expiration, what happens is, first of all, the diaphragm relaxes, and the, see, we have internal intercostal muscles, right? External intercostal muscles, so they contracted. Now we have to go for internal intercostal muscles. Internal intercostal muscles, they contract. Okay, whereas external intercostal muscles, they relax. So finally, the diaphragm relaxes and it gets back upward, and the internal intercostal muscles contract, pushing the ribs inward, thereby collapsing the ribs. That leads to expiration. So what's our option? Internal intercostal muscles contract and the diaphragm relax. Option C is the right option for 186. So another match the following we have here, and we have column 1 and column 2. In column 1, it says true ribs, cervical vertebrae, cranial bones, and vertebrochondral ribs. And we have the column 2, 14, 7, 8, 6. Oh, this must be the easiest question out there because the answers are right before your eyes. So what are true ribs? The true ribs are the one. See here, we have sternum in the front, and then we have vertebral column in the back. Correct, right? So if a rib, okay, if a rib connects both sternum and the vertebral column and it is independent, it is called as true ribs. For example, if I take the structure of a rib, so it's like this, right? Just as a representation, okay? So you can see that in one end it is attached to the sternum, on the other end it is attached to the vertebral column. So these ribs are called as true ribs, correct? Okay. How many true ribs we have? We have 14 true ribs. How it is 14? Each side is 7 and 7. 7 twos are 14 ribs. Whereas cervical vertebrae. What is cervical vertebrae? Just below your skull, the vertebral column starts expanding until your rib region. Okay. So we have cervical, we have thoracic, we have lumbar, and we have sacral. So these are the regions of our vertebral column. So when we look at the number of vertebral bones that are present within your neck region, it is equivalent to 7. Whereas cranial bones, when it comes to cranial bones, how many bones attach your skull? Your skull is not one bone, it is the series of bone that comes and attaches. We have occipital bone, we have frontal bone. Okay, like that, how many bones we have? We have 8 bones which are coming together and they form this cranium or the skull. Okay, next one, vertebrochondral ribs. Okay, so vertebrochondral ribs are 6. So the 6 of the ribs are attached to the vertebrae. So that is why we call it as vertebrochondral ribs, and they are 6 in number. Okay. So what are the options we have? It's very easy. A1, B1, B2, C3, and then we have D4. Where is it? We have our option B. Okay, very easy, right? So let's go for the next question. Choose the wrong statement regarding urine formation. Very interesting question. Filtration is not selective process performed by glomerulus. A glomerular capillary blood pressure causes the filtration of blood through 3 layers, correct? Next one we have is GFR is in a healthy individuals is approximately 125 ml per minute, true. Next one we have the ascending limb of a Henle's loop is permeable to water but allows the transport of electrolytes actively or passively. Now this is a very curious question. Why? Because we have to talk about the first option. Filtration is non-selective process performed by glomerulus. That is true. Basically, depending upon the glomerular filtration, now the blood is forced into the smaller capillaries where based on the pressure difference, okay, within the vessels and the lumen of the glomerulus, now the filtrate is extracted and it doesn't matter what is there and what is not there. So it is non-selective. That is right. Glomerular capillary blood pressure causes filtration of blood through 3 layers. Absolutely right. Okay, we have filtration slits. So you might have heard about the layers, right? It forms the filtration membrane. Okay, the layer of the blood vessel, and then you have the layer of the glomerulus, and finally you have the capsule with the capsule itself. So that is also right. Next one we have is GFR is 125 ml per minute in a healthy individual. This is also correct. Here, it almost appears correct, but it is wrong. The ascending loop. So you know that we have globe, we have Bowman's capsule, and then we have proximal convoluted tubule, and then we have descending loop, and then it goes to ascending loop. This is a very rough structure of, and then we have distal convoluted tubule. Correct, no people? So here, when we talk about descending loop, I'll write DL, descending loop, so it is permeable to water. So H2O, I'll write it as positive. That means it loves water. Water permeability happens. Whereas in the ascending loop, no water gets in or out. So it is highly impermeable to water. So they have given here it is permeable to water. So that is why our option D is wrong. This is a very important thing that you have to remember when it comes to descending loop and ascending loop. Why? We don't have specialized proteins, okay, which help in, removal of water. There are, we call it as water pumps. The water pumps are present in the descending loop, and the water pumps are absent in the ascending loop. Okay, so that makes a difference. Okay, we have one more match the following. We have column 1 and column 2. Enzyme catalyzes breakdown without addition of water. Enzyme catalyzes the conversion of an aldose sugar to the ketose sugar. Then enzymes catalyze transfer of electrons from one molecule to another molecule. Enzymes catalyze bonding of two components with the help of ATP. And we have the names of enzymes. There's an isomerase, oxidoreductase, ligases, lyases. So we know that enzymes are categorized based on their functions, right? So here the functions are given and the enzymes are given. Let's see what are those. Enzyme catalyzes the breakdown without addition of water. Breakdown means what? Is it liation or it is ligation? So for this, you have to just remember your DNA replication. So we call it as molecular gum. Okay. What do? We use ligase. DNA ligase is used for attachment, right? So lyase means what? Breakdown. So enzyme that breaks down is called as lyases without addition of water. So that is why we'll call it as A. Next one. Enzyme catalyzes the conversion of an aldose sugar to, see, whenever it comes to conversion of aldose to ketose, it is an isomerization reaction. Okay, the geometrical isomer is done or performed on the substrate. So that is why it is isomerase. So I'll write it as B. Got it? Next one. Enzyme catalyzes transfer of electrons from one molecule to another molecule. Transfer of electrons from one molecule to another molecule is called as what? Oxidoreductase or oxidoreduction or redox reactions. So I'll write it as C here. Now finally, enzyme catalyzes bonding of two components with the help of ATP. We have seen in DNA replication. So that is D. That is ligase or gum. So what is our option here? We have 1, 2. Okay. A is 4. Correct. Yeah. A is 4. And then we have B is 1. A is 4, B is 1, C is 3. Yes, correct. C is 2 is given here. C is 2. And then D is 3. Okay, correct. Perfect. So we have option C as the correct option for 197th question. So our final question, 199, diabetes insipidus, very curious question, is a condition which a person is unable to produce sufficient levels of hormone, ADH, antidiuretic hormone. The hormone increases the permeability of water of the second convoluted tubule or the distal convoluted tubule and the collecting duct in the kidney nephrons. What is produced as a result? Large volumes of concentrated urine, large volumes of diluted urine, and then we have small volumes of concentrated urine, and then we have small volumes of diluted urine. So usually in the normal condition, what happens? We need antidiuretic hormone. So antidiuretic hormone, what it does is it helps the water to move from lower okay, let's say a less hyperosmotic region to high hyperosmotic regions. Okay, so this happens. So that leads to recovery of water. So when the water is recovered and pushed back to your blood, the urine concentration will happen. Okay, it's just like your filtrate has more water, and we are trying to take out that water and put it into the blood, okay, or into our system. So this naturally happens. So when the antidiuretic hormone is absent, so this process is cut off. So that leads to less water recovery. So that leads to the less filtration rates of water. Finally, lots and lots of urine. See, the people of diabetes, they usually have dehydration, so they keep on urinating very frequently, which is a sign of diabetes. But this is a false diabetic alarm. That is why we call it as diabetes insipidus, where it's not diabetic condition, it is the problem with hormone where a person will not have enough ADH. So that results in frequent urination. And the frequent urination, if you take the sample and analyze it, you see that the urine is highly diluted. So because of more water is present in the urine because of the absence of ADH. So large volumes of dilute urine is formed. Excellent, right? So thank you, students. Okay, we have solved this AIET 6 question. If you have any doubts, please inform your teachers or just put it in the comment section. We'll look into the questions and try to solve. Have a wonderful day and see you in the next video. Take care.